Hi, and thank you everyone for joining today's seminar, special seminar. I would like to begin by acknowledging the Wurundjeri people of Kulin Nations as the traditional owners of the land on which our Parkville campus stands. I also acknowledge the traditional custodians of various lands on which you are all joining me from today. I pay my respects to their elders, past, present, and emerging, and to the Aboriginal elders of other communities. I'm Bhavika, a Center PhD student at Monash Notes. Before starting the seminar, I would like to remind you that the seminar will be recorded and will be available on the CCMP website. Um, just some housekeeping rules that the presentation will last for 45 minutes, followed by 15 minutes of Q&A session. So please save the questions to the end of the seminar. You can also raise your hand or pop the question in the chat box, and I can read that for you in the end. And please stay muted throughout the presentation. Today, we are thrilled to have Dr. Alexey Rak. He is the head of the Center of Excellence and in Structurology and Biophysics at Sanofi in Paris. Alexey brings a rich background in biology, genetics, and biochemistry to his current role in drug discovery. He earned his PhD in biochemistry and biophysics, specializing in protein biosynthesis machinery. His subsequent work at Max Planck Institute at Molecular Physiology in Germany, where he advanced from postdoctoral researcher to group leader, focused on vesicular membrane trafficking. This research earned him a recognition, including the European Young Investigator Award in 2004. In 2007, Alexey joined Sanofi in Paris, where he has made significant contribution to the field of biomolecular interactions, characterization, and protein structure determination. His expertise in advanced techniques like cryo-electron microscopy has played a key role in lead discovery for challenging protein targets. And honestly, I feel truly honored to have had the opportunity to work with Alexey and incredible BSB team at Sanofi during my recent internship. So for today's talk, Alexey will be talking about rational evolution, how cutting-edge biophysics and structural biology are accelerating modern drug discovery. So welcome, Alexey, over to you.
Thank you very much, Bhavika, and thank you very much to you guys for this invitation. So it's quite early morning today here in Paris. My voice may sound strangely, but for those who do not know me, it's okay. I'll be talking today about the interplay between biophysics and the structural biology, and maybe trying to share with you that we don't see very often a border somewhere between the methods. It's just what they're delivering, what pharmacological relevant questions they're answering. We'll see how it goes. Please ask me questions on the way if you like. And then I hope we'll have a discussion about it. So as Bhavika mentioned, we are here in biophysics and structural biology department in the scientific platform, what we call integrated drug discovery, trying to move forward the rational understanding of drug discovery for all modalities. And first of all, before I start, I wanted to acknowledge the team. So here is our team. The photo was taken a few years ago, but still we are as many people as in this picture. And I wanted to thank on the languages that people speak in our perimeter of structural biology and biophysics. So I'm sure that you are not new to the field. Most of you are working in structural biology, and maybe not necessary. Just to remind that the discipline of structural biology, it's quite a young discipline. It started in the mid of the last century. People usually recognize it with the discovery of the structure of a double-stranded DNA, which was done by X-ray crystallography, followed by the first structure of a protein being myoglobin. And these days, we are capable of determining much more complex system like ribosome here that is allowing us to look into details of atomic resolution and explain on that level various biological processes. So these days, we are working with quite complex systems. So here you have a multi-specific drug that is interacting with the two receptors sitting on the membrane, and structural biology can really help to optimize, first of all, recognize the mechanism of binding, then explain the mechanism of action, and give a hand to rationally optimize the drug, making it much more potent and, let's say, more appreciated by the patients. I'm sure that you know that structural biology, in fact, it's sort of a power that is allowing to simplify the processes, difficult processes. I'm sure that you know that most of drug discovery processes in the recent past were driven ligand-based. That means empirically, not having sometimes a good understanding, but a good guess how the small molecule, synthetic molecule, what we call, or large molecules are interacting with the target. So when we apply in structural biology, the process is becoming easier to execute. They are both time and cost effective. And what we do, we are visualizing what's going on in the system. And we are doing it regardless of modalities. So Sanofi is the company that is very generous in terms of various modalities. I think officially announced that we are working on 15 different modalities. And we were just looking that we are supported by structural biology 13 out of 15 modalities. And these days we are very much accelerated and empowered by artificial intelligence, of course, the generative artificial intelligence, but also various tools that be developed and that are accelerating the processes. So why we are doing this? I think you guys who are working with the GPCRs may appreciate, but may not know this paper. Paper is very old. It was published in 2011, and this is a review that is dealing with approximately 1,000 projects that at that time were supported by structural biology and not supported by structural biology. Those who were not supported with structural biology are GPCRs ones. So you may know that the first structure of a human GPCR was determined approximately this time, 2011, by Vadim Cherezov and Ray Stevens in Brian Kabilka lab, for which Brian Kabilka got a Nobel Prize in 2013. So that means it's fair to compare the, let's say, an impact of structural biology to these projects. And you can see it clearly that structural biology enabled projects have a greater chances of success to the market approval than those where structural biology was not applied. So in fact, this is quite an interesting conclusion that was made many years ago, but it still holds true. And this is the reason why we are applying structural biology. So we want to make our projects more successful. So with structural biology and biophysics, our main contribution to the drug discovery is structure-guided drug discovery for biologics and small molecules. We're dividing in this way major modalities. That means synthetic molecules, that means chemical matters, and then also drugs which are proteins. Molecular interaction recognition, so this is about biomolecular interaction, small molecules interaction with the target, or large molecule interaction, characterization of a power interaction in affinity characterization, in the kinetic parameters, but also thermodynamic parameters. We also do fragment-based drug discovery, that means from A to Z, owning the libraries developed and tailored for drug discovery of the starting with the fragments and then doing the primary screening and the validation and growth of the molecules up to lead-like molecule where it can already start its classical way in drug discovery. And of course, the mechanism of action characterization that is done also by structural biology. What we are trying to do, we are trying to be very early in the project. That means if we consider our path from start to Is there a question? No, okay, sorry. So we are trying to contribute to the space which we call pre-M0. We have 3 milestones here dotted in green. M0, this is the start of the project. Sorry, is there any question? Okay, thank you. So we are trying to understand the target. That means with the structure, we are arriving at the early stages, and in fact, structural biology and biophysics contributing with the feasibility, technical feasibility assessment. If we can provide already the structural biology information where the molecule interacting or how the mechanism of action can be used in our processes, that is already a big hand, and that is the case for the small molecules projects, and in large molecule projects, usually the molecules come in a little bit later in the processes, but our main contribution is the structure of the target, gene to structure process, and then ligandability assessment by structural biology and biophysics. So along the value chain, we are also doing by structural biology various characterization. It comes down, in fact, to the structure determination of the complexes drug with the target, and then the characterization or proposals how to improve it. Various other contribution also go into the preclinical studies and the clinical. So we're contributing to the phase 2, to the phase 3 studies, and even sometimes our recent publication in Blood, where we are doing the stratification of the patients also by structural biology. I'm sure that you know how complex is the tool for structural biology and biophysics. We're practitioners. These days, sometimes people are mixing up structural biologists with in silico people, with those who are capable of using alpha fold, one fold, Rosetta fold, and other folds to create the structures in silico to predict it using the pattern recognition, let's say, based methods, not physics-based methods, not Leventhal paradox solving methods, but the methods which is based on the structural biology information and the sequences analysis, which is sometimes very precise in the predicting of the structure. So we are those structural biologists who are doing the experiments, and to do experiments, we need the recombinant protein. So we start with the idea for recombinant protein production, construct design. We go through the expression of the proteins and the purification of the proteins. If that is successful, we try to crystallize it. And if crystals diffracting well, the X-rays, we are collecting the number of images, diffraction images, and processing those can lead us to create a structural model. With the structural model, we can explain the mechanism of action for instance, small molecules, where it fits, what are the interaction, measuring, estimating, and if it's a fragment, growing it, optimizing if it is small molecule, looking at the mechanism of action. For instance, if it's a binder, it can be inhibitor, or it can be an activator, or it can have a potency for blocking the interaction or changing the conformation. So not everything what we need, and we can in fact recognize it with the structural biology on different level. When we're looking, is there a question? I see Ryan is asking a question, right? Ryan iPhone.
I think he got yeah, no.
Okay, good.
Sorry to interrupt.
Very
No
sorry.
problem.
I thought I was on mute.
No problem. So all we are looking at the interaction of biologics on the surface of interaction of antibody, let's say, with our target, and then we can recognize some voids. And these voids, in fact, attracting our attention to try to replace it by amino acid residue side chain that is helping to improve affinity. We call this process affinity maturation, or sometimes affinity modulation, because we also have projects where we need to reduce affinity, for instance, for the brain penetration. And that can be done much easier by structural biology. We are proposing a few mutations, and it's much shorter than to try to generate a bunch of mutants and trying to check them empirically. We're using a lot of biophysics. So to name it, it's NMR. NMR, we're using protein-observed NMR as a structural biology tool, but also a lot of use for ligand-observed NMR. Most frequently, we're using STD NMR, water-loxy, and these days 19F NMR. So this is for the interaction of a small molecule characterization. SPR, so everyone knows what is this, so very high throughput method. If you don't have problems with the conformational changes of your protein upon the binding, so it's something to use, but keeping in mind that you have your protein immobilized and not always SPR is the best method. MST, microscale thermophoresis, where we are very proud because we were at the very beginning of this method and contributed to its development. Hopefully I'll have time to show you some examples. This is the method of characterization of interaction in solution. We are using various thermal shift assay methods, mass spectrometry methods, sometimes also ITC, and many other, especially exotic methods. So these days, as you may know, there is a revolution in structural biology, and that is leading us to not need anymore a few milligrams of the proteins, but sometimes a few micrograms of the proteins. Once it is there, we don't need to go through the bottleneck of a structural biology by X-ray crystallography, like a crystallization, which cannot be guaranteed and may require a lot of construct optimization, a lot of also crystallization themselves, a lot of tests of optimization of the diffraction and then diffraction experiments. So we can now go and prepare the grid. So these grids can lead us to the structure determination and the quality, and the resolution is quite similar to what we can achieve with X-ray crystallography. So this is all possible due to the advances in cryo-electron microscopy. I'm sure that you know, on left-hand side here, you have a protein with the resolution structure determination, which was possible in the last century. Electron microscopy is not a new method. The first electron microscope was commercialized before the Second World War, but it was providing this potato-like resolution, which we cannot use or make using in drug discovery. In drug discovery, we need the coordinates of atoms, and that was possible with the advances in cryo-electron microscopy, a method that was recognized in 2015 as a method of the year. In 2017, 3 individuals who contributed the most to the technology development got a Nobel Prize. And we are using it since then and really appreciating. So here I'm putting a short video. You can see here Anand, who is today on the call. And just to let you know how we try to make it easy, the manipulation and the preparation, the way of a structure determination by cryo-electron microscopy. For Bhavika, it's sort of a reminder where she spent 3 months with us this summer. So sample preparation, Anand is making the preparation of protein sample, and we'll check it by thermal shift assay. So this particular case is a nano DSF. So we are measuring the intrinsic fluorescence. This allows us to find the conditions under which the protein looks more stable, and this is in fact, in our understanding, a good correlation with the success and structure determination. So Eduardo is preparing the for freezing the grids. So he's now pouring the gas of ethane in a bath of liquid nitrogen. So there is a liquid of ethane that is formed, and that will be used to freeze the grids when they're plunged directly in the liquid ethane. So the sample is coming from gel filtration. We usually use fresh preparation under gel filtration. So the sample goes directly to the grid. I'm sure that in this audience, we don't need to explain the principles of cryo-electron microscopy, just to simply show you the simplicity of our operation. So the machines here are used all commercial. So this is the VitroBot 4. We are like you waiting for VitroBot 5 now for 3.5 years since it was announced. Hopefully we'll get it next summer. So when the grids are prepared, they need to be clipped and then also organized to cassettes so that the cassette can be already loaded to the microscope. So this shows how Camille is preparing the clipping and then putting the grids in the cassettes. And Yoshin is looking and collecting data on our microscopes, and once it's successful, already can explain various structures and playing with the structures. So this is a Mickey Mouse way of presenting the whole way, and I'm sure that you guys know how difficult it is. I'll be showing you a few examples, a few projects, and I'm extremely happy that one project, at least that I'll be showing, and a few data were generated by Anand Kumar, who is also present today on the call. So I'm sure that most of you, if thinking about drug discovery, thinking about small molecules, synthetic molecules, and here the first example that I'll bring into you will be about biologics. So on the biologics, what we see, what modalities we recognize, these are the antibodies. These are bispecific and trispecific antibodies, nanobodies, multispecific nanobodies, antibody-drug conjugates, nanobody-drug conjugates, peptides, vaccines, gene therapy, lipid nanoparticles, and others. So with the rational structural biology approaches that we apply, we can explain the mechanism of action. We can do the structural basis for protein engineering. That means based on the structure, it's rationally can be analyzed and modified in a way so that the drug can become much more potent. And we also do the developability assessment. That means risk mitigation. Early in the project, we apply in different biophysics, trying to characterize the proteins and try to filter those which do not look like capable of become a good drug. For us in drug discovery, it's very important to use our resources in the right way. That means if we can kill the projects, if we can kill the drugs earlier, it is very important also for the patient and for our capabilities to put resources on something that is making better sense. With the structural biology, we're also creating multi-specific formats. One of those is a co-DV, it's crossover in DV. It's a tetraspecific drug. We have a few therapeutic proteins in our portfolio based on this. And in fact, the entire format was created using structural biology in our lab. So here we will be talking a little bit about CRCAM5 and the CRCAM5 story. The structure generation by cryo-EM had, let's say, quite prehistory. Prehistory is that we were trying to crystallize and solve the structure by X-ray crystallography since 2014. A lot of resources was put, and only by cryo-electron microscopy we could determine it. Here in red, you can see post-translational modifications, which in fact was possible to see on the structure. And one of those, in fact, is the part of the epitope that is recognized here by the Fab. Fab is a part of that antibody that is used in antibody-drug conjugate. So I'll not tell you the full story. I will only tell you something that I think makes sense. So CRCAM5 has 7 extra cellular domains, Ig-like domains connected in the knuckle. And here they're all very flexible and they're highly glycosylated. And this was the difficulties, let's say, broken by structural biology with this. I'm sure that you know what is the ADC. ADC stands antibody-drug conjugate. So you have the antibody that is delivering the drug, and the drug is conjugated usually to the Fc part of the antibody. The drug is a cytotox molecule or is specific to a target, let's say, drug. And the idea is that the antibody is recognizing the receptor, which is specific for one or another type of the cells, can internalize, and then the drug can be released directly in the cells that either we want to kill or we want to bring some effect using this drug. So in fact, here we are talking about the DM4 drug, and this is the drug that is shown on this picture. So this drug was developed for non-small cell lung cancer. The size of the molecule was already not very optimal from the beginning. We knew that the interaction lies somewhere on 2 domains, A3 and B3, that is recognized by the Fab of the antibody, and that is altogether making only 68 kilodalton. So I'm sure that you know that there is a limitation in the size. Although now we are also trying to prove that with this limitation we can deal. We just got accepted our paper in scientific reports where Camille Samson in our lab determined the structure of a target of 20 kilodalton. That means we publishing the way how to increase the size of the molecule in an easy way that can lead to high resolution. So here is an example of using different microscope, the 300 and 200 kiloelectron volt, and you can see that in the region where we had the size of our molecule, there are not so many examples. And that time we had a 200 kiloelectron volt that was used to generate the data. In fact, we tried to work with the full-length protein. We tried also many other constructs. We tried to fix the ectodomain by diabodies, triabodies, and something that really worked due to a lot of efforts that Anand applied was only this small complex. So at the end of many trials, we could already see something that resembles the Fab and also the target, which was in the complex with the Fab. And that in fact led to the structure determination. So you have here in yellow the CICAM5, the two domains, and in fact between those two domains, or closer, let's say to the B3, you have the Fab, and in Fab, as you know, you have light chain and heavy chain, and they are interacting with the protein. So here we have on the left-hand side an epitope. That means the part that is recognized on the surface of the target, and on the right-hand side you have a paratope. And the connecting, let's say, part or touching part, they are highlighted here in pink. So you can see here on the left-hand side that the part of the glycan is in fact also a part of the epitope, which is difficult to imagine to generate this data using X-ray crystallography. For X-ray crystallography, we are dealing with we have better chances to crystallize when the proteins are homogeneous. That means either we are mutating these parts or these amino acid residues that can be glycosylated, or we are reducing the glycans to the last fructose enzymatically, trying to make the sample homogeneous so that it can be prone to be crystallized. So here is an example where cryo-EM is representing an opportunity, therapeutic opportunity, working and characterizing highly glycosylated samples. So in fact, we had some data, previous data using HDX. HDX, as you may know, stands for the hydrogen deuterium exchange method. So this is the mass spectrometry method where the samples can be put in the solution of deuterium, and those hydrogens which are surface exposed will be replaced by heavier deuterium, and by mass spectrometry you can do the analysis of the complexes and also of the proteins alone, and that can lead you to identification of the interaction. So here on the left-hand side you can see the results of HDs of epitope mapping, and on the right-hand side you have a cryo-electron microscopy. So you can see that there is quite a difference. Of course, on the right-hand side, you have real structural biology, and on the left-hand side, this is on a structural model, the understanding of interaction based on a biophysical method, HDX. So if we are looking at some data that was also early available and not structural biology, that was ALAScan. ALAScan, this is the method where all amino acid residues, in this case on the paratope, that means part of antibody, were replaced one by one by alanine. All mutants that were generated were probed for the interaction with the antigen by SPR, surface plasmon resonance, which is determining the affinity, and based on the outcome, in fact, the paratope can be mapped. So you can see here that it's quite accurate if you're comparing to the experimental structural biology by cryo-electron microscopy on the right-hand side. So this is an example where we were dealing with a suboptimal complex. So 68 kilodalton complex was a determined structure using 200 kilovolt electron microscope. And at the epitope paratope region, we reached to 2.6 angstroms resolution. We characterized it. And it was interesting that the epitope paratope interaction aligned with the alanine scanning to some extent, and to lesser extent with the HDX data. So what we have these days, we have the process that is allowing us to determine the structures within days, what before was sometimes requiring months. So here we have an example of a target that we determined the structure. It was also literature unprecedented heterodimer, and we put approximately 2 years to determine its structure. More than 200 constructs were generated since it's a heterodimer. All those were probed for crystallization. Many constructs, including mutations, optimization of surface entropy, because the crystals were not diffracting to our satisfaction, and approximately 1 million investment was done. Later, when we in fact got it was a cancer target for oncology program, and there was a generation of cancer resistant mutants, and we wanted to see how the structure of the protein may look like. As you know, AlphaFold or any other predictions not providing you with this information. It will not predict the conformational changes upon single mutation, point mutation. So we had to do it experimentally. So that means it's that rare case where we could compare how this 2 years and 1 million investment can be a bit now by cryo-electron microscopy. And again, this is an example of work done by Anand. So Anand did collect the data within approximately 2 days. That means preparation of the sample and then data, and that led approximately to 3 angstroms resolution on the 5th day. There are also some examples when the structure can be determined within approximately 4.5 hours, A to Z. That means collecting the data, processing this data, and already having a model to be described, answering pharmacologically relevant questions. So these days, we have talents, we have a hardware, we have software, and know-how how to do it. We have a multi-sites presence of structural biology. Mostly our center of excellence is located in Paris here. Here we have 2 microscopes, so Cryos G4 and Glacius, both with energy filters and a Falcon 4i. We have also 1 microscope in Lyon and 1 in Boston, and now we also got Cryos 5 that will be installed in Waltham in the Boston greater area. So having this distribution over the sites, we at the very beginning, when we tried to put in place cryo-electron microscopy back in 2020 internally, so we created an access to the cloud. So we don't have any on-prem solution. We don't have a storage. So all data that we are generating by our microscope, they go on the cloud. On the cloud in Magellan environment, we have the installation of all our software. That means all our software were converted to global licenses and everyone can access it. And for instance, the data that can be collected in Paris can be available to our colleagues in Lyon or in Boston for data processing literally next second. And that is allowing us to have a good teamwork and innovative, let's say, moving for the structure determination. So very interesting question here. You can see this very nicely aligned operation using cryo-electron microscopy. And the question based on what I presented to you, the comparison of cryo-EM with HDX, can be structural biology replacement by physics. So this is what we can see. So here I put time and the consumption of the material for various methods. So here we have the HDX mass spectrometry, we have the ALA-scan, we have X-ray crystallography, and we have a cryo-EM. And what we have on the left-hand side, we have a protein consumption, data acquisition, data processing, data analysis, and elucidation. So these are the main stages. And we were trying to project, how long does it take, how much it costs us, because we have here to count for cost effectiveness, for time effectiveness, and then to choose the method that can be the most, let's say, competitive for us to drive our drug discovery projects. If we don't have a problem or, let's say, bottleneck with the crystallization, X-ray crystallography would be the method of choice. As you know, these days the data acquisition takes approximately 2 to 3 seconds, definitely in 5 seconds. You have a 360 degrees on goniometer head of a synchrotron. The data are processed on fly, so this is much older method, and much more efforts were put to create working flow. So this is what we are dreaming for cryo-electron microscopy, have data processed on fly. And in a few hours, in fact, you can have the structure determination with the full analysis. For the HDX, you need, let's say, 100 approximately micrograms. You need 9 hours, 3 replicates, few hours for data collection, and the data analysis takes days. ALA-scan to generate approximately 40-point mutations. It's weeks, sometimes 2 months. With the high throughput that is now developed, it can go down to a few weeks, but this is still, we are talking about weeks, not days and not hours. So in a cryo-electron microscopy, effectively, we need 1 to 5 micrograms. That is the amount that we have on the grid. We may need more for, let's say, trying to identify the best conditions. It takes approximately 15 minutes to a few hours for the data collection. I'll show you an example of those, and a few hours for the data processing. So maybe also a few hours for the analysis. So definitely here one can see that the cryo-EM can be a method of choice. But what is even more important, if you are doing ALA-scan or if you are doing HDX or any other biophysical method, or biochemical biophysical method for the characterization of epitope and paratope, you may always look for a validation by X-ray crystallography or by cryo-electron microscopy, because HDX or ALA-scan, it's a good guess. It's educated guess, it's informed guess by experimental method, but this is still not a structural biology. While if you are having already reached the structure determination by cryo-EM or X-ray, you would never be looking for HDX on ALA-scan, because this is the experimental method, and usually it's already answering your questions. So here we have an example of, again, by Anand generated. This is also suboptimal sample in the size. The size of this complex is approximately less than 70 kilodalton. The data collection was 26 minutes, and the data processing 3 hours 40 minutes. Most of this is the motion correction, and that led to 3.5 angstroms. And this is the resolution at which we can clearly, with no ambiguity, already can see the interaction, all side chains, all amino acid residues which are involved in the interaction. Next is a little bit better in terms of resolution. Anand was able to deliver the average 2.92 angstroms. That means much better resolution on the surface of interaction. And it is due to the fact that this sample is already better optimized in terms of the size. These are the 2 Fabs that are interacting with 1 antigen from 2 sides. Simultaneously, we could form this ternary complex, and in fact the data collection was taken only 14 minutes, and the 3.225 minutes was for the data processing. So we are 41 minutes, and I'm not sure that I have time to tell you about small molecules. So I'll be passing, and maybe we'll be talking to you sometimes later. What I wanted to tell you is that for the small molecules, we have various methods that we applying for drug discovery by fragments. And we identified that the best primary screening that we are doing is by cryo-electron microscopy. That means the hits that we can identify that can lead to a molecule that we can optimize into a drug. We invested quite early, let's say in the 19F NMR. By NMR, we were lacking the method for determination of affinity that we, in fact, developed and published some time ago. And more recently, after already putting in place the new way of 19F NMR in drug discovery, we published a paper that is now available for everyone in methods molecular biology this year, explaining how we are doing it. And we are trying different methods. I missed the part where we were contributing to development of thermal shift assay by nano-DSF and MST. But all the time we are trying to find a drill or a tool that can in fact lead us to the structure determination. And in fact, trying to see how MST is delivering or how the NMR is delivering, how it is correlating with other methods. Can it lead us to the molecule of our interest? But in fact, at the end, what we need the results. We need the holes. And this is why we are now back to X-ray crystallography for cryo-electron microscopy, for X-ray crystallography for screening of the fragments. I'm sure that you know what is the X-ray crystallography and how important is this method. But in the context of a high throughput method, we in fact can replace the classical methods by something that can make it faster and can be adopted to drug discovery. And that faster and adopted to drug discovery is the high throughput in terms of the preparation of the sample, which is not new for the plate setup. What is new, a little bit different, is that we are doing the soaking experiments using ecosystem that is allowing us to do the approximately 96 soakings in 2 minutes and generating many crystals that is allowing to improve the high throughput these days. So the working flow that is described here, very quick screening of the fragments, was established by Yulia, our postdoc in the lab. And in fact, with this automated working flow, we can do it better and faster, the project support, but also one of the new applications is a primary fragment screening by X-ray crystallography. That means no needs for the screening of the interaction, but doing it directly by soaking. And there we have a dedicated library, which is characterized, let's say, by high solubility, so that the soaking can be done at approximately 50 millimolar concentration. So with cryo-EM, definitely we are extending the application, and extension of application for integral membrane proteins, in fact, is enabling us in drug discovery and rationally deal with the targets. As you know, many targets in our organism, in human, is the transporters, GPCRs, SLCs, and these days we can do it by cryo-EM. And doing it by cryo-EM, that also implies a big difference in the way we are producing the proteins. Not anymore needs to produce a large amount for X-ray crystallography, but a small amount that can be assessed and then scaled up if needed. I'm sure that you feel that X-ray crystallography and the cryo-EM best friends in structural biology these days. I think those who are using both having a better chances to success and sustain the drug discovery projects in the company. Talking to you, I cannot avoid touching a little bit on AI. So the big AI because, small AI, machine learning and bioinformatics are used in structural biology since its inception, because we are processing the data. Based on the processing of the data, we are learning something. And these are the big data, usually by X-ray crystallography and later by cryo-EM and interaction characterization. But these days, when the generative AI was put in place, there is a significant changes that in all three major parts of a cryo-electron microscopy, where you have a sample preparation, where we have a data acquisition, and the third method is the data analysis. We have AI that is contributing. So in fact, the AlphaFold, the OneFold, RosettaFold, others, they help us to do the construct design. Once we have the model, it's allowing us to do the X-ray data phasing for unprecedented proteins, protein structure. And for cryo-EM, it also helps to elucidate the initial data. So we are extremely happy that in 2021, when it just arrived, AlphaFold, our first presentation was a Nobel Prize worthy methodology, and it's people got a Nobel Prize for that. So nowadays, for the competitive cryo-electron microscopy, we need data acquisition, which is made also using AI, what we are doing, then also data processing and analysis using various tools based on AI algorithm, and then the structural data elucidation and interpretation. So what we have these days in Sanofi in terms of a cryo-EM, I'm sure that the cryo-EM that is of your major interest for this audience, we have 5 instruments. We have, in fact, everything what is allowing us to characterize to get the structures for the project support. What today we are doing, we're even generating the structures to feed our AI tools. That means the publicly available artificial intelligence generative tools, they don't have maybe enough data, or the data not allowing to be as precise as we require. So in fact, we are generating sometimes data internally to fill the gaps in the metadata, and that is allowing us to make the tools working on specific, let's say, targets for us better than what is available outside of the company. So what we are still dreaming about the revolution happening in sample preparation. There is a resolution revolution, but unfortunately not in the sample preparation. These days, if we are talking about cryo-electron microscopy, it's the latest stages of film photography. It's very fast to go to supermarket to get your prints using your film, but it still takes time. You can see you cannot react quickly as in digital photography, whether the picture you took is good, the sample behaving well. There is still a delay. I showed you a few examples where within hours Anand was capable of determining the structure, but it's still a few hours. So our dream is to get a digital cryo-EM, and we do believe, with all experience that is accumulated for X-ray crystallography during the last 70 years, can be used to press down the time that is requiring. And one of our dream that we are moving to is imaging of the cells. Tomography, in a way, as it is known now, is not really something that we can apply because the size of our subjects and because of the resolution that tomography can deliver. But we are trying to play with a different technology so that we can look at the details of interaction on the membrane that would allow us maybe revolutionize the biological discovery. And that all goes to the way to deliver the drugs faster. And I think with that, I'll finish it here, and hopefully I'll have your questions. Thank you very much.
Thank you, Alexey. That was amazing. Also, cryo-EMs solved a lot of problems for structural biologists. So yeah. I would like to open the floor for questions. If you have it. We have one question. Could you please comment why we need to lower the antibody affinity in order to penetrate the blood-brain barrier? I had this question too.
Yeah, so you can read, there was a few papers. I think the pioneers in this field were Gene and Tech. And they showed the interaction with C receptors and the difference and dependencies on the percentage of the brain penetration and the affinity. So sometimes it is the case. We can confirm it on a number of our, let's say, examples and the projects. There is definitely a difference whether, you always have to do it. I'm not sure, but sometimes you simply need to find a tool to modulate affinity. That means being able to increase affinity and decrease affinity. So that's what I wanted to mention.
Perfect. Thank you. And Matt has one question, so go ahead.
Hello?
Yeah.
I don't know if my camera's working. It doesn't look like it. But anyway, so I guess, it's very noble to have, a structure really fast, and that's great. But what about quality? Because I mean, the maps that you showed looked incredibly over-sharpened and almost potentially uninterpretable. So I think that there has to be a balance between quality and interpretability and spending your time doing it properly. So maybe you could speak to that a little bit.
Yeah, absolutely. So it's a very good question. I think it's not only about the quality, but also what you sometimes understand and the quality, the resolution. And in
No,
drug
I don't think
discovery,
resolution is a good measure of quality. I think things like how the Gurney air plot looks, how B factors are sharp, how B factors are attenuated at higher spatial frequencies. These things are really matter, especially if you want to exactly lock in, say, small molecule drug positions and determine this accurately. I mean, this is incredibly important and takes time.
yeah, absolutely. We absolutely agree with you. And the example we showed you on biologics, in fact, this is where, let's say, the answer, the structures can be generated quickly. With the small molecules, indeed, we have to collect much more data. And that is leading to more lengthy processes, but still, we never release any information or any description where we cannot judge without ambiguity about positioning of a small molecule. The same quality assessment applied internally as it is done externally. So when we are, for instance, publishing, we are not brushing up the internal structures. So they are going, if it's X-ray crystallography, through WATCHECK and PROCHECK and other procedures. So that is telling about the quality of the structures that are kept in internal database. So, but what is different, and that's not only about the resolution, but also about the quality, is that we are goal-oriented. That means we are not in a position to spend much more time to playing, maybe to push the resolution or to push the quality if already unambiguously with what we have in hands, we can answer the pharmacologically relevant question.
Yeah, and maybe just a follow-up question too, if it's all right, Viveka. So you talk about digital EM. I mean, it already is. And look, the major problem with direct electron detectors is Poisson noise, which is impossible to overcome due to the fact that we've got to image at low dose. So I guess I want more clarity about what you mean by digital EM, because in my mind, it already is.
Yeah, so digital is like a digital. I made it digital photography. That means you took a picture and you see what is good on the picture. If you don't have what you have in your frame, you simply retake the picture because you need to prepare a sample differently, or maybe you don't have a complex that you're looking for. So now between you are taking the images and the time you see what is on the images, there are hours of data collection, there are hours of data processing. So we want to make the moment taking the picture and seeing the picture shorter. Of course, digital is the aim. We do understand that there is a limitation, an instrumental limitation and the technology limitation, but this is definitely what we would be dreaming of.
Oh, cool. I don't see any question in the chat box, but I have a question. So for this entire workflow of 3 hours thingy, how many micrographs are you taking in the beginning? Like how many raw data? Like how many micrographs
Yeah, so
to reach to Armstrong?
Anand, do you want to chime in?
Yeah, I can. First of all, this is our default process to quality control. Whenever we get something new, we always go through initial small data collection to just check the quality of the data that we want to continue for bigger data set. And for these two examples, one of the example we only collected 140,000, 140 images only, and we got 2.9 angstrom. And I can say that these maps doesn't look really good because they are directly coming from Cryospark without sharpened at all. But if you use tools like Deep EM Enhancer and things like that, they could be clearly nicely visible. They are intended to show like low resolution weird, but if you use tools like Deep EM Enhancer, they can be really um, nicely resolved maps.
Oh, we have another question from Meenakshi.
Hi, Alexey. I hope you can hear me. It is an amazing talk. I just have a very general question, might be a naive. When you go for, like, let's say you collected a data set and you got like 3.5 angstrom, and then you collected another data set on the same sample and you got up to, let's say, 2 angstrom. Would you mind commenting on this? Like, is it dependent on the sample quality? Or have you experienced anything that maybe you used a different type of detergent or something different in the second sample that helped you get into that high resolution?
Yeah, thank you very much for the question. I mean, actually, so in fact, there are many factors that are contributing to the quality. Quality also translates to higher, let's say, resolution or details that we can learn directly from the resulted, let's say, structures. Well, first of all, what we are dealing with is the suboptimal size. And if within the suboptimal size, we also have some flexibility, we are definitely either to collect much more images or we are to optimize and working with the sample detergents, different type of molecules which are not detergent, let's say, or working with, for instance, the nanodiscs and the SMALPs and different other polymers that are used for membrane proteins these days really screened, and sometimes the difference can be tiny, either concentration of the same detergent or combination of a few detergent that can lead to the improvement. Of course, the construct on its own, that means what we have in a sample, if we are able to try to fix it by preparing a new construct, making it less flexible. With that less flexibility, do I lose the information that can be relevant to the answer to, let's say, mechanistical questions about the changes of the conformation. So there are many factors that are available to improve, but there are also many no's that sometimes are preventing us to do it. We are doing it, but with that we are losing something. So we always have to find somewhere a compromise, working with the sample that would make sense to us, getting the resolution and the quality where we can feel self-confident when we are commenting about the structure.
Thank you. Thank you so much. I really appreciate it. Thank you.
I don't have any question in the chat, and I think we are in time, just right on time.
Good.
So thank you so much, Alexey. I guess I don't know what seminar is happening next, so I don't have any announcement to make, but I'm so happy you came and gave us this seminar. Thank you so much.
Thank you.